Hello, and thank you for subscribing to my Substack. I'm going to do something new here. This is my first video post, and the topic is what makes us human puts us at risk, brain evolution and mental illness. What this talk concerns is what patients often ask us every day in the clinic. How did this happen? Why does my child have autism or bipolar disorder or schizophrenia? These are some of the most painful conversations that clinicians can have with their families. And I think to answer these questions, we need to really take a deep look at brain evolution and how some aspects of mental illness arose from that evolutionary process. So let's get started. Humans are an evolutionary success story. As you can see in both these graphs, the human population has grown exponentially. That's particularly true since the invention of agriculture. And yet, as you'll notice on the right, the world population growth is declining, and in fact may become negative by the year 2100. There are several projections of population on the right that show the human population could actually decline. Of interest, as you can see on the left, that decline is particularly marked in the industrialized countries, Europe and Asia. The American population in the United States is still growing, but that has for many years been attributed to the immigration, both in terms of immigration increasing the population and the fact that immigrants were generally having children above the replacement rate, so that we may see the USA population turning negative in the very near future. Many people argue with the state of the environment, maybe that's a good thing. The world can't support that many people. But in fact, if the majority of our population are elderly and need support from younger people, and the number of younger people decline, that could be an issue. One of the intriguing facts here is that most mental illnesses generally decrease the fertility rate relative to the general population. This is particularly true for schizophrenia. Surprisingly, it's also true for bipolar disorder, even though people in their manic phase tend to be hypersexual. As any clinician knows, most patients with bipolar spend most of their lives in their depressive phase, which does indeed have hyposexuality and decreased fertility. People with autism rarely have children. And interestingly, people with ADHD do have a fertility rate higher than the general population, but that tends to be offset by an unfortunate increase in the risk for early death, usually due to accidents or other illnesses related to lifestyle. So we have this paradox in which generally throughout the world, psychiatric disorders are increasing, and yet those very psychiatric disorders should, why did they not sort of phase out of the population? Now, this leads to certain thinking, well, maybe these psychiatric disorders are positive in some way. We're going to look at that issue in some detail. First, let's look at some basic evolutionary concepts. Natural selection is the differential survival and reproduction of individuals due to differences in phenotype. That is, does a change in the phenotype lead to an increase in reproductive success for the population as a whole, not for each and every individual? So basically, genetic variation creates new phenotypes. Those phenotypes could be beneficial, they could be deleterious, or they could just be neutral. And we need to avoid some kind of Lamarckian or often it's called teleological thinking, that there's a reason behind evolution. So while we may tell our 4-year-old that the giraffe has a long neck because he reaches for the leaves, it's not reaching for the leaves that made the giraffe's neck longer. Random variation created giraffes with short or long necks Those with longer necks could eat more and survived, thus passing their genes on to their offspring. Neutral traits that are neither beneficial nor harmful are simply randomly sorted in the population. So there's a myth that every trait that humans have must be there for a reason, or there's something good about it. For instance, if you feel on your earlobe maybe either goes straight across or maybe it's notched a bit. That trait is under genetic control. But it's randomly distributed in the population. There's no advantage or disadvantage to that particular shape of your earlobe. And also critical is the concept of the evolutionary trade-off. So a giraffe, in order to extend his neck, they must have very high blood pressure so their blood can reach that elevated head. And that required the evolution of a new cardiovascular system. So really it was not enough for evolution to select for a long neck. It had to select for a long neck and for this enhanced cardiovascular system. And sometimes things play off against each other. The best example of that is sickle cell trait. So the heterogeneous state is an advantage wherever malaria is endemic. The population survives longer. But being homozygous for the trait means getting sickle cell disease. So a quarter of the population may suffer from a terrible disease and even die early, but the trait continues because in this heterogeneous state, it's beneficial to the population. The evolutionary trade-off is key. So, and key, a deleterious trait must reduce reproduction to be bred out of the population. If the trait has its impact after reproductive age, such as risk for dementia or Huntington's disease, that gene will remain in the population. All right. Now we have some pernicious myths here. One, if something's genetic, well, it's permanent, it's invariably passed from one generation to another, and genes determine the behavior or the disease. All right. That's clearly not true. There are many environmental interventions for a genetic disease. In contrast, if a risk factor is environmental, we then think, well, the trait's malleable, just change the environment. And in fact, environmental effects through epigenetics can cause permanent change or damage to an organism. This debate tends to be polarized in the right and the left. Sometimes the far right will say, oh, everything's genetic, that's just the way it is. The far left will say, no, everything's environment, we just need to change the environment, when in fact the gene by environment will be critical, as we shall see. Now, here's a good way of understanding genetic risk, because I think a lot of people are still hung up on Mendel's peas. You know, you get the gene for the round pea or the wrinkled pea, and that's it. So imagine two tables. If a hammer falls on your toe, if it falls off the table, the hammer causes your broken toe. Now, think of the genetic risk as to where the hammer is on the table. So if you have low genetic risk for the disease, it's as though your hammer is in the middle of the table. So when the environmental event occurs, bumping the table, you are less likely to get the disease. The hammer is less likely to fall off the table. In contrast, if you have high genetic risk for a disease, that hammer is placed close to the edge. So if the table's bumped, highly likely that the hammer will fall off and break your toe. So a small environmental push may push you toward the disease. We can prevent the disease by addressing the underlying genetic risk. That's very difficult. Or we can alter whatever environmental effects is, being careful not to bump the table, for instance, in this analogy. Now let's go directly to human evolution. Here we need to go back many billions of years. The Earth has gone through many ice ages. The last ice age was, the last warm period was quite prolonged. About 65 million years ago, the asteroids killed the dinosaurs, and that allowed the mammals to emerge. And not long after that, the Earth descended into an ice age, one of which we're just coming out of now and maybe coming out of very quickly due to the effects of climate change. The first bipedal apes emerge as this ice age increases about 8 million years ago. Now, if we take the last 400,000 years and blow it up, that's in the lower part of the figure, we can see that the first Homo sapiens emerged about 300,000 years ago. But then our ancestors, modern Homo sapiens, really emerged in Africa about 65,000 years ago, left Africa, populated the rest of the world, and also triggered the Neolithic revolution that led to advanced tools and agriculture. More on that in a moment. First, let's go back quite a distance, and here I would recommend the text A Brief History of Intelligence by Max Bennett, who breaks down the evolution of the brain into numerous categories that were critical. And first he points out that the evolution of animals occurred about 650 million years ago, and 550 million years ago the first bilaterans appeared. That is, animals that were symmetrical. They had a left side and a right side. Most of them were tubes, as is the most classic example of this group, the nematode C. elegans, and that is shown, that creature is shown on the right there. This creature is of great interest. First of all, this basic plan, having a symmetric body, is 99% of the animals alive today. And the chief advantage is that being a tube allows you, and being symmetrical allows you to steer. You can go right, left, or up or down. C. elegans has a small brain, actually has two brains, but we'll focus on the one in the head, 302 neurons. It has dopamine, which it uses for approaching food and getting food when it's hungry, and it also has serotonin, which helps it determine when it is full. So C. elegans is in the water or the dirt. He eats, defecates, and has sex. Some might say that C. elegans has the meaning of life all worked out, because we are basically also a long tube. The difference is we have a much larger cortex perched on one end of it. Now, as I said, while mammals first evolved about 300 million years ago, they really didn't come into their own until after the dinosaurs died and their niche opened up. But what made the mammals particularly special? Well, again, looking at Max Bennett's review of things, of course, compared to the fishes, the mammals developed a much more complex neocortex. But what exactly did that neocortex do? And Bennett focuses on the role of the agranular cortex. Agranular because of the way it looks under the microscope. As you can see on the left, the little dot neurons are not there, cells are not there, hence it is agranular. And in humans, we find this in parts of our motor cortex and somatosensory cortex, as well as in the anterior cingulate cortex and the supplementary motor cortex. Well, what does the agranular cortex do? Well, on the right, you can see that it actually has very little input, very little direct sensory input. That is done by the granular cortex, which is everywhere else in the brain. So information from the sensory parts of the brain is moved into the agranular cortex, where it is processed, and in Bennett's theory, or his review of the literature, mammals are able to simulate the future. So the little proto-mammal that's under a rock can look out and see, oh, there's some food, I can go get it, but there's a predator who might eat me. If I go now, I'll be eaten. If I wait till the bird is gone, I can get the food. The mammal can simulate the future and thus engage in much more adaptive behaviors that lead to the future. And what do we say as humans to each other? We say, think about what you're doing. When we say to our children or to our patients with ADHD, you need to think about what you're doing, we're talking about this process of simulating the future, because then you are likely to make better choices if you can run through the different options. Now we get to our primate cousins, and primates consist of the lemurs, the monkeys, and the apes. And we are part of the great ape family. Now here's interesting, the apes have multiple complex social arrangements. So the gibbons, the lesser apes, they are monogamous. They mate in early adulthood. They stay together for life. They raise the child and remain together after the child moves on. The orangs are solitary. They live by themselves in the jungle, come together only to mate, and then the mother raises the child on her own. The chimpanzees and the gorillas have the classic alpha male orientation. There's a senior male who kind of controls what goes on in the troop, usually by physical strength, although in the case of the chimpanzees, the alpha male must have alliances with many of his younger male friends who help him maintain that position. And in return, he shares food and females with his allies. Now, in contrast, the bonobos are very different from the chimpanzees. They have no real hierarchy. Males and females are equal. They are pansexual. They tend to resolve conflicts by having sex with each other. And interestingly, the male bonobos remain with the mother, and the female bonobos go out and start a new troop. Now, we humans can have aspects of all four of these arrangements, plus many of our own. The basic point being is that the kind of social engineering of the brain was already there, before humans evolved. Now, another intriguing thing is that if you look, is the concept of sexual dimorphism. That is, how much are males bigger than the females? And the more the males are bigger, the more you get a alpha male, quote unquote, patriarchal social arrangement. You can see with chimps, the males are bigger, not so much in the bonobos. Now, as bipedal apes evolved, interestingly, the Australopithecines, they sort of enhanced the sexual dimorphism. But that's not the route that evolution ultimately went. Instead, we kind of moved back toward what the bonobos did, in which there is only minimal sexual dimorphism between males and females. Now again, I need to emphasize we're all part of the great ape family. So here we see men sharing a beer, a common human experience. Over here on the right, we see male chimps sharing fermented fruit. So the yeast and the fruit are in a symbiotic relationship. The yeast gets sugar from the fruit, makes alcohol, and in return, animals come and eat the fruit as a source of feeling good and also of calories. So here we see the alpha male and two of his bros sharing this fermented fruit. And you can almost see the sense of satisfaction even in this pre-verbal species. You can imagine them relating to each other. Hey, remember when Louis got chased by the lion and the lion almost ate him? Oh wow, that was great. This is we with the great apes, as part of the great ape family, share this intimate social interaction. So why bipedalism? Well, first, our evolution is not a straight line. As I say, here go the Australopithecines, and I'm not going to say a lot about them, but they were really just bipedal apes. That was kind of it. But it didn't work out. We went in a different direction, and a lot of different things had to happen. And what were those things? Well, first of all, a major reorganization of our skeletal structure, a major redesign. Why was this an advantage? Well, as the Ice Age accelerated, the African rainforest turned to savanna. That gave an advantage to creatures that could walk or creatures that could run. You could cover more ground. More food, If you're more bipedal, you can stand on a tree branch and reach up and get what's left of the fruit as the forest begins to thin. So that becomes an iterative process that over millions of years, bipedalism is an advantage. But that wasn't it. We also became hairless, we developed hands, and most importantly, we developed large brains. So to be bipedal creates what has been known classically as the obstetric dilemma. So if we look in the upper left, we can see that as a result of bipedalism, the human pelvis narrows, and it is much more difficult for the infant to get out. And on the lower left, you can see how this particularly became an issue with Homo erectus and then finally with Homo sapiens. And indeed, maternal mortality, death during childbirth, remains a very serious problem throughout the world. And even today, with modern obstetrics, there's many, many dangers. Now, in the upper right, you see the core of the problem. If there is a wide birth canal, then the pregnancy cannot be supported very well because the whole weight of the fetus is falling on the muscles of the pelvic floor. In contrast, a narrow birth canal may be good for the pregnancy, but massively increases the risk of obstructed birth. So how did humans solve this problem? Well, they did it through midwifery. Midwifery requires an intense relationship between the mother-to-be and the midwives. The midwives must be part of a community. So communal relations now become even more important than they are for chimpanzees and bonobos. In the upper left, we see fertility goddesses, it's thought to believe. It's unlikely that at the height of the Ice Age there were very many people around who looked like this because food was in short supply. But early Homo sapiens was aware that maybe a wider berth would mean easier birth, that breasts were needed to nurture the baby, so that became an idealized view. And there are sort of rub marks on these statues which suggest that perhaps people held them, maybe much the way people will hold a rosary or a cross today. On the lower left, you see from the Bronze Age also some figurines of the birth canal, but these appear to be much more technical. You can't see, though, it's hard, you have to disassemble them to see the depiction of the birth canal, almost suggesting that these were educational aids, just as we give our medical students anatomical pieces today to look at. And finally, on the right, we see a sculpture from 300 BC from ancient Rome, and this looks very modern. These could be modern-day obstetricians. We see the one woman holding the baby, another assisting, and a mother delivering the placenta. Again, a whole community has to be around the mother. Humans do not give birth unassisted except in the most extraordinary emergent situations. And we always have had the profession of midwifery, which of course today is the profession of obstetrics. Now, this recently has been the source of fascinating genetic studies. All of these are genes that govern the structure of the human pelvis, and particularly the female pelvis. And down in B, you can see pelvic width, pelvic inlet width, and so on. To what aim is all of this going on? Well, here we see the evolutionary trade-off. Again, here are the different genes with their different spots on where the gene is on the chromosome, and we find that evolution has constrained female pelvic width within a certain range of the bell curve. If we have a larger birth control, we have increased risk of pelvic floor failure, as well as pelvic floor disease, which is very serious. We can have increased risk of hip osteoarthritis. In contrast, at the other end, the smaller birth control increases the risk of obstructed labor. and also problems with the knee and the lower back. And of course, with the advent of modern obstetrics, somewhere around the 1840s, anesthesia, forceps delivery, and finally cesarean section, it is much less risky for women with a narrow birth canal to give birth. And so we would expect that evolutionary constraint to be somewhat relieved. But we're going to look at this more in terms of there's always an evolutionary trade-off. A good thing often is coupled with a not so good thing. Now, one interesting modern consequence with change in environment, female athletics, particularly soccer and volleyball, basketball, are much more enhanced in the last several decades. It's become much more competitive, much more aggressive, and puts much more strain on the body, more similar to the way that male sports have been. And of course, many people know that ACL ligaments have increased over the years. And it's clear that this may be part of the evolutionary trade-off. Compared with males, as we see on the left here, females are at higher risk for sport-related injuries due to wider hips, shorter femurs, and more hip internal rotation during movement. This is part of that tendency to shape the pelvis to make birth easier. Also, it's not every female athlete that's at particular risk. There are, as we can see here, if a young woman carries a certain genotype, there's a gene by environment interaction which further increases her risk for ACL injury. And those collagen gene variants that are associated with ACL injury are also associated with joint laxicity. So genes that make it easier for the pelvis to relax and allow the baby to emerge also put one at risk for ACL ligaments. Now, what we see here is a key point. Ways to decrease ACL injury include strength training, balance exercising, and stretching routine. In male sports, there's been massive investments in kinesiology over the last 50 years. Anyone who looks at an NFL or college athlete now can see that, to reduce the risks of various forms of injury. There need to be similar investments, knowing this vulnerability, for female athletes so that they are less vulnerable to a ACL injury. All right, coming back to evolution, again, I have problems with this diagram because evolution is not a straight line. Homo erectus is really a different paradigm from the Australopithecines, but one of the key points is that as we move from Homo erectus to Homo sapien, we become hairless. What is that all about? Well, except for humans, all mammals thermoregulate by panting. In contrast, we have no hair and hence we can sweat. And here we have the tail of the head and pubic louse. So these are different species of lice, and they separated from each other 1.5 million years ago. One lives in the head hair, one lives in the pubic hair. And 1.5 million years ago is roughly about the exact time that Homo erectus emerged. So that tells us that Homo erectus was hairless, and we also know from his pelvis and leg bones they are just like us, so he probably ran. So Homo erectus was out doing the marathon, chasing after the big game, spearing it, probably hunting, and for that we know social skills must have been needed. Furthermore, that meat eating means that a greater source of calories is present. Now, despite losing our hair, we Homo sapiens have great attachment to our hair. Our babies will cuddle with puppies. They enjoy that. We give our infants hairy mothers to artificial hairy mothers. And if we don't do that, our young will sometimes make it for themselves, as the child on the right has a transition object. That is a smelly, hairy object that's very comforting to them. And of course, while emergency management people beg folks not to send teddy bears to a site of a disaster, because they just have to be thrown in landfills, It is a human impulse. I see people in pain, I will comfort them by giving them a fuzzy mother, a teddy bear. And the existence of teddy bears goes long before they were named for Theodore Roosevelt. And of course, another issue that confronts humans is that our ape cousins and monkey cousins use their hair to transport their young. Here's two young chimps on their mother's back. Here's a howler monkey baby riding on her as she leaps through the jungle. Of course, our hair hardly supports our infants. We must carry our young on our backs, and I assure you these people will not be smiling in 5 minutes, even if this weren't a stock photo. We often are in conflict with our infants over their hair, our hair, because they instinctively try to grab it and hold on to it, and that's one of our first social interactions. No, no, don't hurt mommy, ow, ow, don't pull. The beginning of inhibitory control. Rather, we must make tools, whether that is a sling, a papoose, or here as you see a $7,000 stroller. Well, to make tools, we need hands. So we can look at hands as part of the package that needs to go, it's coevolution. We need to be bipedal, we need to lose our hair, we need to start eating meat. Well, to do all this, we also need our hands. And here I'm going to introduce the concept of human accelerated regions in the genome, and they're usually abbreviated by HA or HAR, before the gene name. And we have, our hands are different from the chimp hand in two critical ways. We have the precision grip at the top, which is most classically associated with a baseball or cricket ball. We also have the power grip, where we can swing a hammer, swing an axe, or swing a club or handle a sword if we're going to be aggressive and assaultive toward our fellow Homo sapiens. The ability to do that is the result of this HACNS1 region, which, as you can see, is strikingly different in humans relative to all of our other mammal cousins. And you can see how this emerged gradually as the hand becomes more nimble from the chicken all the way up to the human. Now, the other thing that's going on here, so we have this creature that's a little more big-brained. He's running, he's hairless, he can hunt, he's out on the savanna. What's happening on the savanna? Well, now the savanna is drier. We don't have the rain every day. And thunderstorms, lightning can set fires. And brush fire are a natural part of the savanna ecology. So surely Homo erectus was observing fire. He probably saw animals that were burnt in the fire, smelt it, tasted it, said this is pretty good, and could start by bringing fire back to a campsite and lighting a fire, and probably not long after that learned how to make it on his own. Maybe, as in far left, even setting fires in order to drive animals in a certain direction to make hunting even easier. So cooking becomes part of the human repertoire. Well, interestingly, the work here that you see in this journal looks at how the human genome changed with cooking. So the chimpanzee has a very large colon that they need to break down the high fiber plants that they meet. And while human brains use 20% of their daily intake for the brain, the chimp only does 9%. He has to devote a lot of their metabolism to the gut. Overall, humans have a much higher total energy expenditure than the chimps, even adjusted for body size. So those calories come from cooking, whether it's cooking meat or cooking vegetables. We have evolved, from 2016, it was clear that we evolved a series of liver-related genes that assist us in particularly processing cooked food. More recently, you and colleagues have looked at gene expression in various animals versus humans, And what you're seeing here is genes in humans that are differentially expressed in us versus chimps or mice. And you can see that there are some genes, the M1 category, that we express less, and others, M2 and M3, that we express more. And some of these are interesting. Not surprisingly, a lot of them are related to protein digestion and cholesterol metabolism, a disease that we now suffer from since our food availability, particularly high-fat food, is so much more available. Now, look down here, spinocerebellar ataxia. What does that have to do with eating? Well, nothing. But maybe in the process of bringing on these genes that are so beneficial for digestion, this was an evolutionary trade-off. A small group of people have this disorder, but the population as a whole benefits. Doesn't mean that cerebellar ataxia is somehow a positive thing. So here we go. Not only is a ice age coming, the temperature's getting colder overall. Winter is coming if you're a Game of Thrones fan. But during the last period, the climate is fluctuating wildly, as you can see here. Some years very cold, some warmer, and that means flexibility, ability to change your routine, learn from experience, becomes much more selected for. And that is when we suddenly see the big increase in brain size, certainly over the last 500,000 years. So we have Homo erectus, a brain nearly as big as ours, but still smaller. Very adaptive, left Africa, populated Asia and parts of Europe, ultimately died out. As far as we can tell, no genetic contribution to modern humans. And but then anatomically modern humans emerges 300,000 years ago, but lived very much like Homo erectus. But then about 80 to 65,000 years ago in Africa, people who were very much like us today emerged. These were Homo sapiens that clearly had an advanced culture. They made art, they wore jewelry, they painted things in their caves that obviously had symbolic meaning to them. And how did this occur? Well, one theory is that Mount Toba in the Indonesian islands had a massive eruption. That is certainly true, but that maybe created a global winter for many years, and perhaps that killed off or started the die-off of early Homo sapiens and allowed the most adaptable people to emerge in Africa and now spread out to the rest of the world. That is debated. Some people say you just can't get the correlation that tight, but nonetheless, that is one theory. So modern sapiens comes out. When they reach Europe, they run into the remnants of the Neanderthals and the Devassians, another group, early Homo sapiens in Asia. And they intermingle with them. These are not different species of humans as we had once thought. And there's a gene flow between them. So modern Homo sapiens, we're not some kind of pure race. We in fact have always intermingled our genes with others. And eventually, both the Denisovans and the Neanderthals died out or were absorbed into the Homo sapien line, and we spread to Europe, to Asia, and ultimately to Australia and the Americas. Now, today, we have 2 to 3% of our genes from the Neanderthals, maybe a smaller amount from the Denisovans. Most of those are immune genes. So what we gained from the Neanderthals was the ability to fight off the infections that the Homo sapiens from Africa now were confronting in Asia and Europe. But there are some other genes that's very interesting. For instance, genes for smoking, the risk for smoking, genes for sleep problems, genes to consume alcohol. If you get more Neanderthal genes, you're slightly more at risk for those disorders. Well, that's odd. We know the Neanderthals were not smoking. They certainly weren't drinking alcohol. But one thing the Neanderthals were doing is they were in caves at the height of the Ice Age, making fires, breathing in smoke. One of the things that the cholinergic genes that put you at risk for smoking do is they make you more likely to tolerate the adverse effects of smoking. Anyone who's tried smoking earlier in life knows the first time you smoke it, it's like, God, this is grotesque. And people who have that reaction say, I'm not doing this anymore. But some people, this is not good, but it's not so great. But boy, that nicotine hits pretty good. So maybe thank you, Neanderthals, for giving us those smoking genes. Now, more interesting, there's a curious phenomena that if you've got certain Neanderthal genes actually are protective in a very minor way against most major psychiatric disorders. Now, the one exception to that is ADHD. The more Neanderthal genes you have, the slighter your ADHD risk is greater. So this argues against the idea that, let's get rid of the idea that the ADHD gene is a hunter gene. That's just a bad idea. In fact, clinically, I can tell you a specific example. Practicing in Texas all my life, I'm very familiar with hunting culture. It's a rite of passage for fathers to take their sons on the hunt. And I had a father I treated his son's ADHD, and he thanked me profusely. He said, now I can take my son with me on the hunt because he now can listen to instructions. He's no longer running around making noise and scaring the deer away. He said that before he was treated without his medicine, if he handed him a rifle, the only thing that would be safe in the forest would be the deer. So we can see that it's pretty obvious that hunting, particularly hunting with a bow and arrow or a spear, requires quiet and concentration and focus. So ADHD is not a hunting gene. More about that in future substacks. All right, so chimp puberty begins around 7 to 9 and ends at age 13. Of course, human puberty usually doesn't begin until later childhood, early teens, and ends around, in terms of endocrine changes, around age 16 to 19, but CNS maturation doesn't usually end to age 25. And by that I mean, in particular, the pruning of the gray matter and the growth of white matter, which ultimately leads to advanced executive functions. There is evidence that the Neanderthals entered and completed puberty earlier than the modern humans. So now we get to the heart of the matter, human accelerated regions. These are human-specific genes that appear to trigger our large brains. What do these HARs do? Well, here we see that these three areas, the HARs, they're conserved in the orang, the gorilla, the chimp, but they are unique. They are altered in humans. And so here we can see the chimp brain. Here's two genes. They're separated from each other. Humans have an HRI inserted in here, and even though these two genes are identical in the human compared to the chimp, this structural change means these two genes interact with each other and thus change their gene expression. The proteins are expressed at a different time and in different amounts, which leads to changes in brain function. And that is how we can have 98% of our genes shared with chimps and bonobos and yet still be so different. So how was increased brain capacity achieved? With apologies to Sir Mix-a-Lot, the key point here is I like big brains and I cannot lie. So a quick review of genetics. We have two major forms of genetic variation. The single nucleotide polymorphism, or SNP, where the nucleotide pair is switched and there's no structural change to the DNA. And then we have the copy number variant, in which areas of the DNA are either duplicated or deleted, so sort of the length of the DNA changes. With SNPs, they have effects on what I call normal variation, range of IQ, personality dimensions, attention, anxiety. Symptoms are kind of in the normal range, but when they become extreme, problems emerge. Whereas CNVs are very much related to the major mental health disorders, as well as to physical anomalies that tend to be associated with various syndromes. But these CNVs had very positive effects in early human evolution, which led to our big brains. So let's walk through these. Now, don't freak out. There's a lot of terminology here, but I'll walk you through it. So the FOXP2 gene has a 2 amino acid substitution in it that makes us different from the other great apes. Neanderthals have the sapiens version, but there's evidence that the regular process is changed so that FOXP2 is different in modern Homo sapiens relative to Neanderthal. People with mutations in the FOXP2 gene are very rare, but they do have language disorders. But FOXP2 plays a very wide range in synaptic plasticity, big brain development, and critically, motor control and coordination. So the fact that humans can do this may be partially related to FOXP2. We can also go A, I, O, U. We can make vowels. We can manipulate our vocal apparatus very minutely. That too is partially contributed to by what FOXP2 does for us. And evidence of that is even in other animals, FOXP2 has influences on their ability to do communicative vocalization. But please do not think of FOXP2 as some kind of single language gene. So here's FOXP2. It's regulated by various transcription factors. It's big in transcriptional repression and chromatome modeling. That is, it controls other genes. And some of the key genes that it controls are right down here. Let's walk through these. A fun one is SRPX2, or sushi repeat containing protein X-linked, named sushi because that's what it looks like in its chemical structure. Take my word for that. It is critical in developing the glutamate synapse. CNT-NAPT is one of the largest genes in humans. It's involved in cell adhesion proteins and above all in neural glial cell interactions and cerebral cortex development. It's less methylated, that is, it is more active in humans. All right. Now it is thought to be associated with many psychiatric disorders. Disrupted in schizophrenia 1 was named long before we knew what its role is, but it is highly involved in brain development. There it is right there. AUTS2, once named for autism before we knew what it does, in fact is its official name now is activator of transcription and development regulator 2. It's a master regulator influencing the processes of neuronal differentiation, migration, and synapse formation. As a result, you can see that how impaired neurite outgrowth would be highly related both to autism and developmental delay. Next, we have the Notch family, little known but important structures that kind of create these protein bridges between the pre- and post-synaptic neuron and thus enhance their communication. Humans have 3 functional copies of this gene, whereas other apes have only 1 functional copy. And mutations here can cause microencephaly, macroencephaly, and developmental delay. Finally, humans have a version of a gene, OLIG, which governs the white matter development. And humans have many more copies of it, and this leads to not only greater white matter, but greater white matter plasticity, enhancing human learning. All right. So all of these things are positive in early evolution, but it's a little tricky. You have them, big brain, only takes a little to go wrong, and we may have increase the risk for serious mental illness. Now let's take a look at the SNPs. To understand these, we need a quick review of a concept called heritability, which is the measure of how much of the variance in a trait is related to genetics. So the heritability of height is very high, 0.9. That means, let's say we have a generation that is raised in poverty, and as a result their mean height is relatively low, but the height within the population shows the normal human variation, some short people, some tall people. We improve the lives of these people, and for their children they're all taller than the parents. But because heritability is high, the rank ordering of the people is the same because genetics governs the variance. So the tallest father still has the tallest son, shortest mother still has the shortest daughter, and so on. In contrast with IQ, heritability is only 0.5. So again, in the impoverished population, IQ may be somewhat low on average, but some high IQ, some low IQ within the population. In the next generation, education and the general lot of people has improved, so the population is smarter overall. We still have smarter parents having smarter children, but the rank order is not as strict. This is because the environment is controlling half of the variance. So gene by environment interaction becomes very important. All right, so when we look at the genetics of IQ, there are 205 spots on the genome that are associated with IQ in the latest study I could find. These are all the dots that are above the dotted line, and this involves as many as 1,000 genes. Not surprisingly, these are all genes that are expressed in the brain. We're only accounting for about half of the heritability that we think is related to these SNPs, as you can see here. The gray is the missing heritability. All right? And not surprisingly, genes that control the growth of certain areas of the brain are associated with intelligence. And not surprisingly, that's the frontal cortex is involved, but so is the amygdala and the hippocampus, the accumbens involved in reward learning, and surprisingly, the cerebellum. We don't use our cerebellum just to do this. We probably use the cerebellum for a lot of intellectual activity. Now, the genes that are correlated with high IQ are also negatively correlated with ADHD, obesity, and coronary artery disease. Now, that does not, I want to emphasize not, does not mean that people with ADHD or who are overweight or have coronary artery disease are less intelligent than other people. It means that there's an evolutionary trade-off. Genes that help the population be more intelligent also have a tendency to put one at risk, to negatively put one at risk for ADHD or obesity or CAD. Now here's another important point. If we take the population and we divide them into different socioeconomic groups, they range from the wealthiest people, that's the first, to the poorest people, that's the fifth. What do the green bars represent? They represent the number of, for lack of a better word, IQ genes that convey the probability of a higher IQ. Surprise, surprise, the poorest people who are very bright, they have more of the good stuff. The complete opposite of the idea, well, people who are successful in society, they must be super people. They must have more of the good stuff. That's why they rose to the top. We found this study kind of found the opposite. That the people with high income are getting a lot of good environmental input to raise their IQ, whereas the people in the deprived environment, the more likely way they get to the higher IQ is they do have more of the IQ genes to begin with. Now, this study by Shaw in colleagues that was published in Nature back in 2006, I think has not gotten the degree of attention that I think it should. So hopefully I will give it some attention here. This is a population of individuals who had serial MRI scans from early childhood to young adulthood. And you can see that on the x-axis in each graph. And on the y-axis is cortical thickness. And let's focus on the top two graphs because they show the pattern most clearly, or even on the top right. Let's look at that one. We're dividing the three groups up by the level of their IQ. So people with average intelligence, have a certain cortical thickness at baseline, and you see that you normally prune your gray matter, over time, and it reaches a slightly lower level at age 19. Well, for the people who are above average IQ, just a little bit smarter, they have more gray matter to start with, but they kind of parallel the average group. They prune their gray matter, and they're still a little above the average people at age 19. Now look at the people with superior IQ, the genius IQ people, if you will, using that term they actually start out with less gray matter. And suddenly they grow their gray matter very rapidly, peak a little later, and then they prune more rapidly. And curiously, they end up roughly at the same level of the average people. And yet, of course, they're still very high IQ. So it's not just the absolute level of cortical thickness, it's the pattern of brain maturation that's critical. All right? Well, here in the ENIGMA study, which has looked at thousands of people in terms of their cortical thickness and the relationship to disease, there are two kind of major findings from those studies. And you can find this by searching for the ENIGMA study. You can find this diagram as well as the explanation behind it. Notice in schizophrenia, there's a great deal of gray matter loss, particularly in the frontal cortex, which makes one think, is there excessive pruning? And in autism, in general, not in everybody with autism, but in certainly significant numbers of people, we find an overgrowth in the cortical areas. So these two processes that produce high IQ people, they also put people at risk for two of our most serious neurodevelopmental disorders. Now that does not mean that people with schizophrenia are geniuses or people with ASD are geniuses. In fact, people with ASD are also at risk for intellectual disability. Cognitive deficits are common in schizophrenia. We're struggling to find treatments for those. But it suggests that there's an evolutionary trade-off going on. All right, hold that thought because I'll be coming back to it. Now I will shift gears to sort of the more about the SNPs and normal human variation. There is a test called the Big Five personality test that looks at normal variation in personality. And this test looks at openness to experience. That kind of correlates with risk tolerance, how likely you are to try something. Extroversion, being very sociable. Conscientiousness. Neuroticism, agreeableness. And we have found SNPs are associated with, but our missing heritability is very high, whereas genetics accounts for half the heritability. We've only found around 5 to 8% associated with the SNPs, as you can see in the across here. This shows how they're correlated with each other. Usually not. These are fairly independent, but obviously the people who are very open also tend to be extroverted and so on. All right. My main point here is that SNPs are associated with these personality traits. Well, a way to look at it, and this helps you understand gene by environment interaction. So let's say there's a score on a trait from very low on the trait to very high. Well, the SNPs are distributed as so. People that have a lot of SNPs for a trait are going to score high, and here's the point at which we would consider it pathological. Now over here what we can see is looking at young adults, people who are age 16 to 39 who took the test recently have lower rates of conscientiousness, higher rates of neuroticism, lower rates of agreeableness and extroversion. Well, nothing's changed in our genome in the last 10 years, but this is the generation that has experienced COVID and other stresses. So what that means is that while the blue dots are the risk for the trait, we've shifted the environment. We've pushed the hammer closer to the edge, the hammer is at a point in the table, but we're knocking the table much more strongly. And as a result, those with less genetic risk are now at greater vulnerability to clinical elevations of this particular trait. Now, well, do you want to get rid of the SNPs? There may be companies that say, yeah, we'll do that for you. We'll measure your SNP risk. We'll do that. I say,
danger, Will Robinson. Danger. No, Will Robinson. Danger. Danger, Will Robinson.
So there's evolutionary trade-offs. We've already done this in our poor animals. The French bulldog, as cute as they are, because they're so inbred, is prone to many very difficult diseases. So the CNVs, large CNVs that are related to intellectual disability, to physical anomalies, to profound autism, these perhaps can be identified prenatally, and that's settled science, I think, at this point. But trying to use a SNP score, commonly called a polygenic risk score, to pick someone, an embryo that's has greater likelihood of high IQ or more likely to be extroverted or to be a musician, that's very dangerous because we run the danger of inbreeding our population. That's not the way evolution is designed to work. So here I'm going to come to my key points. The environment selected for bipedalism, but the survival of the bipedal apes depended on large complex brains that required hairlessness, safe birth, and a series of mutations that not only created large brain size but increased connectivity for creativity and relatedness. Further mutations of these genes, though, are deleterious. They raise the risk for schizophrenia, autism, and other major mental disorders. We cannot breed the genes that make us human out of the genome, all right? They're going to be there. So autism, schizophrenia, we will always have with us. But that does not mean that there are advantages to having schizophrenia or that it's good to have autism. These are unfortunate conditions that we must treat. We don't say, we understand that sickle cell anemia, sickle cell trait had survival value at one time and still does in certain parts of the world. But that does not mean sickle cell anemia is a good thing. We seek out treatments for it. Thus, we want better treatments, new treatments for autism, schizophrenia, intellectual disability. Evolution likes diversity. It selects for personality styles as a whole. Extroverts are valuable because they try new things. Introverts are valuable because they think about problems and come up with solutions. We want a mixture of people. So they value the person but at all times, but separate the disease from the person. And our goal as healers is to decrease the burden of disease, whether it is measles, whether it is cancer, or whether it is schizophrenia. And with that, I hope to see you more on my Substack. I hope to have more videos in the future. Please feel free to put comments or questions. I welcome healthy debate or to expand on some points that maybe are not clear. So I hope everybody has a great day. Thank you.